Hello, everyone. This is my privilege to introduce Dr. Karen Lincoln from the University of Southern California, who will present a talk entitled Biopsychosocial Risk Profiles Among African American and Non-Hispanic White Adults, Findings from the Health and Retirement Study. Her talk is co-sponsored by the PRC and Center on Aging and Population Sciences, directed by Dr. Deb Umberson and Dr. Karen Fingerman. At USC, Dr. Lincoln is an associate professor in the Suzanne Dorak Pack School of Social Work. She has published over 70 articles and book chapters in the areas of stress, aging, and mental health disparities, and is a public scholar and aging advocate. She has op-eds in the New York Times and Wall Street Journal and other media outlets focusing on long-term care and policies to support poor seniors. Dr. Lincoln has testified before the Senate Select Committee on Aging and Long-Term Care. She was appointed to both the California Task Force on Family Caregiving by Senator Kevin De Leon and to California's Master Plan for Aging Research Committee and Equity Work Group by Governor Gavin Newsom. Dr. Lincoln has received numerous honors of her career, which recognize both her scholarship and advocacy. Dr. Lincoln is a fellow of the Gerontological Society of America, a Harvard faculty scholar, an Encore Public Voices Fellow, and a Next Avenue influencer in aging. She is also recognized as one of the most influential scholars in the field of social work. I would like to remind you that during the Q&A following Dr. Lincoln's presentation, we reserved the first 2 questions for graduate students. Please join me in welcoming Dr. Karen Lincoln.
Thank you, everyone. Thank you for that introduction. I really appreciate it. I'm going to do my best to share my screen. Hold on one second. Let's make sure I'm sharing the right thing here. Okay. Just 1 second. Okay. All right. All right. Let's see if we can get this working. All right, you all can see that? Very good. All right, thank you. Thank you, everyone. I really appreciate you having me. Thank you for inviting me to give this talk today. So here, as you heard, is the title of my talk, and we're just gonna get right into it. So just, I wanted to give a general overview of my presentation. One of the first things I wanna do today is talk about my approach to scholarly research, because I think it's really important to understand why I did this particular study. I'll follow that by talking a little bit about what we know and what we don't know in terms of the background for the study. I'll talk about why I did this study, go through the methods results, summarize the results, and talk about a few implications for moving forward. And then I'll talk about future research, both as it relates to this study and as it relates to the research that I'm currently doing. So starting with my approach to scholarly research. So I'm both a sociologist and a social worker. And as part of that, it's really important for me to look at different ways of understanding complex problems. And as a sociologist and being trained at Michigan and ISR, you know, we are taught to look at a range of demographic variables and sometimes some interactions. And we often look at between group differences, comparing whites to blacks, men to women, et cetera. And I really like to look at the heterogeneity within groups. And so part of the work that I do is really taking things apart, right? We're sort of looking inside and seeing what's happening within groups, because I think that's where some of the really important knowledge, particularly as a sociologist and social worker, come from. That is where we find those variables on which we can intervene. And it's very difficult oftentimes when we're aggregating populations together and comparing groups to actually see what's working within groups and what might not be working within groups. So that's one of my approaches. And the other thing is I like to think outside of the box, right? And so I think by being trained interdisciplinarily across disciplines and working with people across many different disciplines, it's helped me to see the world in different ways and understand it in different ways and acknowledge the strengths and limitations of different disciplines and bringing them together and trying to, you know, use the strengths across those different disciplines to my work. And so one example of that is a paper that I did as a graduate student when I was at U of M. And I was very much enamored with Karen Rook's work. In fact, I attribute her seminal piece that she wrote in the 80s to the work that I do today. And as a result of discovering her work, I did my preliminary exam on social relationships. And one of the things that I wanted to do was to take Karen Rook's work and apply it to African Americans. And so this is a study that was published when I was a graduate student. And it sort of featured looking at social relationships in sort of both the positive and the negative aspects, right? The dual nature of social relationships. Up until this time, most of the research was really looking at social support as a positive type of resource and not really paying attention to the negative aspects. And as a result, the findings were a bit equivocal. So there were groups of papers, large groups of papers that showed that social relationships had a very positive impact on psychological well-being. But then there were papers that showed that there was no relationship between these constructs. And then a few that showed there were negative impacts of social support on psychological well-being. And so what I did back then and what I do in my work is I pull them apart, right? You take out the positive from the negative. And then once you do that, you actually see that in fact, oftentimes positive aspects of social relationships do have positive outcomes, depending on the outcome. And then negative aspects of social relationships, in fact, do have these negative outcomes. And so it's really taking things apart, which is sort of the nature of my work. This is another example of sort of taking things apart, looking at both the positive and negative aspects of social relationships. And this is one of the first papers that looked at this sort of dual nature of social relationships and depressive symptoms among older African Americans. And it was published in the Journal of Geriatric Psychiatry. And so the moral to the story is this. If you examine emotional support from family, which is the positive aspects, on mood disorders, anxiety disorders, and the number of mood and anxiety disorders, there are no significant relationships, right? So social support had zero impact on these mood disorders and anxiety disorders among older African Americans, but negative interactions did, right? And so this is really important because if you were to aggregate, right, social relationships, you would get a very different outcome. And so this really shows the potency of negative aspects of social relationships. And so another example of me sort of taking things apart is this paper that was published in Social Science and Medicine. And I was really interested in looking at African Americans and Caribbean Blacks because a lot of the work up until this point was sort of aggregating Black Americans together and not really considering ethnicity within Black Americans. And so this was sort of a wink and a nod to looking at heterogeneity within Black Americans, but also trying to understand the relationship between race and depressive symptoms, right? So there's a lot of literature that reports that African Americans have higher levels of depressive symptoms, but lower rates of depression, right? And so there seems to be this like paradox where you have, really high exposure to stress, really high levels of depressive symptoms, but not quite tipping over into those clinical levels. And I wanted to really disentangle that a little bit. So I did this study and I used latent class analysis. And it's really important to say that now because I also used it for the focal study for today's talk. And what I found were there were actually two subpopulations in these data. And these were I used the National Survey of American Life, I believe, for this study. And it showed that there were two different groups of people in these data. There was a group that had high levels of symptoms and a group that had low levels of symptoms, right? So when you aggregate them together, you're going to get something very different. I went a little bit further than that and then looked at each item, how people in each of these profiles were endorsing each of the items, right, on depressive symptoms. And the patterns are very similar, right? You'll see a little bit of difference. But the other thing that you do with this type of analysis is you can see who's in that profile, right? How are people different? And so here you can see for people who are reporting high symptoms, they had certain types of socioeconomic characteristics and psychosocial factors, right? Lower SES, more women, they were high on negative interaction and lower on social support. What's also key here, particularly with the more women, there were also men here, right? So we get a lot of reports about how women tend to have higher levels of depressive symptoms, but there were men in this group too, right? So this again shows you some of the limitations of comparing groups because there's lots of heterogeneity. For this low symptoms group, there were more men, but there were women as well. They tended to be older. They had lower levels of negative interaction, but the thing that struck me was the high discrimination. I couldn't really understand why people who were reporting low symptoms of depression had high discrimination, particularly when the literature says that high discrimination tends to be associated with higher depressive symptoms. So I wanted to investigate that a little bit. And the other thing I wanted to point out here before I move on is that within both groups, there were African Americans and Caribbean Blacks in both profiles, and ethnicity didn't significantly determine which profile they were in. They were sort of equally distributed between those two profiles. So I wanted to look at depressive for racial discrimination because I was a little bit confused by that finding. So I did a similar type of analysis where you can look at how people are endorsing the items. And for this major discrimination measure that I used, you can see for both groups, the low symptom group and the high symptom group, they had some similarities in terms of the types of racial discrimination they were reporting. The patterns are a little different. And where you see those white dots, those are where the low symptoms group were reporting higher incidences of these types of discrimination. And so what you might notice is that for the low symptoms group who tended to have higher SES, right, and lower symptoms, they were reporting more discrimination in terms of jobs and where they live and in terms of service. So these are more public types of situations where these sort of higher SES low symptoms groups were reporting discrimination. So that helped me understand why the low symptoms group had higher levels of discrimination. It had to do with the particular domains in which they were reporting discrimination. So one thing I wanted to share with you is I was a guest editor for Generations, and this also is an example of my approach to the work that I do. So I was asked to be a guest editor on an issue that focused on economic inequality. And after meeting with the board, we decided that it would be a little bit more like the kind of work that I'm really passionate about. So in addition to looking at economic inequality, I also wanted to look at social inequality. And so we ended up agreeing that we would look at both economic and social inequality. And I also wanted to look at it from a life course perspective. And I wanted to invite people who focused on younger populations, children, to see how, that might actually, some of the experiences that children have, how that might play out in adulthood. And Jackie Angel also contributed a beautiful piece to this guest edition. Thank you, Jackie. But you can see here that in addition to looking at both economic and social inequality, looking across the life course, we're also really interested in what we can do, more practical understanding of what we can do to address economic and social inequality. So there are studies here that talk about, different programs that are actually sort of looking at and addressing social inequality and economic inequality. So basically, my research is driven by this sort of dogged preoccupation with inequity at various different levels, both economic and social and others, and how we can actually close the health gap, right? So the sociologist in me is really interested in those larger societal and social level factors. And then the social worker in me is, okay, what can we do about it, right? There's more of a practical sort of application aspect to the work that I do. So now we're going to move on to talk about knowledge and gaps. And this is sort of what we know and what don't we know, particularly when it comes to race and health. So African Americans have an elevated risk for earlier onset of fatal and non-fatal chronic conditions, right? And so that's not news, right? So of course, this leads to some of our health disparities, right? Both in sort of longevity and health span. And health span is the definition of that has to do more with healthy, active years of life and free of chronic disease, right? So that's sort of the definition of health span. And so several years ago, 2017, the CDC put out this report as part of the Vital Statistics series. And it basically showed that younger African Americans were more likely to live with or die from diseases that were typically found in older whites. Right? And so that includes heart disease, stroke, diabetes, and you can see from this figure that at all levels, even from the ages of 18 to 34, you see these race disparities with chronic diseases that many African Americans are living with at very early ages, right? And dying from, right? They're more likely to die from these diseases as well, even though they can be controlled. And so despite the fact that we know this, we don't really truly understand the causes of these elevated risks, right? We sort of get at it in different ways, but we still have a fairly poor understanding as to why there's this elevated risk among African Americans. Some have talked about accelerated aging, right? So these sort of premature declines in health may be this manifestation or reflection of this accelerated aging process, whereby African Americans are getting older faster than whites in particular. And so there are some studies that have looked at this, and I know Bridget is very well informed about a lot of these studies. And so they're looking at biological age, right? And so looking at biological markers to understand biological age. And these studies basically show, many of these studies show that African Americans are in fact aging faster than whites, right? So there's a study that was done a few years ago by Levine and Crimmins, and they used a whole host of biological markers to understand cellular aging and biological aging. And what they found, that there was this accelerated aging for African Americans. And so what you see here, that across age categories from age 30 to age 89, African Americans are biologically older than whites of the same chronological age, such that the category of age category of 60 to 69, African Americans are more than 5 years older biologically than whites of the same chronological age. So studies like this sort of point to this idea of this accelerated aging process. And so for this study and for other studies, cellular aging is often indexed by, again, a host of biological markers, one of which is the telomere, right? And so telomeres, you'll see here, these are actual pictures of telomeres, which are those little yellow dots, right? To explain more about that, these are telomeres. They're right at the tip of the chromosome. They're DNA-based caps and they're protein structures, right at the tip there. And they shorten with each cell replication. So that's just normal, right? So every time your cell replicates, your telomeres get shorter. As you age, they get shorter. So that's just a natural process, right? But once they get critically short, then they lose their protective function, right? So then you have the risk of long-term cell cycle arrest, you have the risk of cell death, right? So senescence and apoptosis. And so that's where we start to see this higher level of vulnerability to chronic health conditions when telomeres get critically short. And so what the research shows is that people who experience are exposed to higher levels of stress, particularly across the life course, their telomeres shorten faster. And so we start to see, particularly in middle age, a lot of chronic health conditions in African Americans specifically compared to whites because of this sort of accelerated telomere shortening over the life course. And so what these studies have shown is that short telomere length or greater rates of shortening, if they're shortening faster, predict a host of both mental health and chronic health conditions, right? And so there's some, differences in terms of findings. There are equivocal findings, right? And we'll talk a little bit about that as we move through the talk. But for the most part, we're seeing this relationship between shorter telomere length and some of these chronic health conditions that we tend to find in older adults and that we tend to see in African Americans earlier in the life course. And so here's where I come in, right? So we do see some race differences. There aren't a ton of studies that look at race differences, but there are some. And what they show is that early in the life course, particularly at birth, there's no difference in terms of telomere length between racial groups. But when African Americans and whites and other groups start to get older, we start to see some differences. Although there's some studies that show there are no differences between African Americans and whites. There are some studies that show that African Americans have longer telomere lengths than whites. And then there are some studies that show that African Americans have shorter telomeres than whites, right? So it's kind of equivocal. Overall, though, if you look at cross-sectional studies and longitudinal studies, there is some evidence that there is sort of a faster rate of telomere shortening for African Americans compared to whites. So, but there are some knowledge gaps, obviously. Most of the studies that look at race, it's really to document differences by race, right? And because I'm more interested in risk and protective factors, I mean, that's pretty much what I've examined all of my career. I'm really interested in those factors that will help disrupt that relationship between race and death, right? So just saying that, say African Americans, as an African American, I have accelerated aging and I'm more likely to die earlier from these diseases, and just leave it there is not enough for me, right? I want to figure out like where can we intervene? Can we? Is this just so, and there's nothing else that we can do about it? Or are there ways to disrupt this relationship, right? So that's what I want to do with my work. And so this is a study that was published in the Journals of Gerontology, Social Sciences, and I was really trying to inject myself into the conversation. And I stayed with my Bailey Awakened Social Relationships to see, well, are there different types of social resources that might disrupt this relationship? And oh, by the way, are there also racial differences? And so I did this study, and the findings did show that, yeah, social relationships do matter, right? They do in fact matter. If they're positive, it depends, right? How much positive relationships you have in relative to negative relationships, right? Ambivalence. Do you have more positive compared to negative? Do you have more negative compared to positive? But what it ultimately shows is that social relationships matter, and that if you have positive relationships, it can disrupt that relationship. If you have negative relationships, there isn't a relationship between negative interactions and shorter telomeres. It's true for Blacks and whites. The difference is the rate at which negative interactions is related to telomeres is different for African Americans than it is for whites. So it appears that even though both groups experience negative interactions, both groups have shorter telomeres as a result of negative interactions. It appears that that shortening is a little bit faster for African Americans who experience negative interactions compared to whites. So why another telomere study? Some say, oh, telomeres have gone by way of the dinosaur, but I am just preoccupied with a couple of things. One has to do with looking at mitigative factors beyond just as correlates, but as actual sort of intervening factors. And then there's a paradox that I'll talk a little bit about later that I wanted to address with this study. So I do want to credit telomere studies because they have advanced science in terms of understanding how early life adversities in particular, and also the accumulation of stress and other social factors are related to the progression of disease and aging. So they have made their mark. They're very important. However, our understanding of the relationship and the interplay between race, biological and psychosocial factors is really inhibited by the way that some of the telomere studies are conducted, right? And I'm specifically talking about the fact that there's a lack of attention to heterogeneity, not just race, not just ethnicity, but biologically, right? So I wanted to conduct one more study to examine and to at least account for some of those differences and variation within and between populations. And then the other thing that I wanted to address is that there are all of these different interactions that people experience, right? So at the individual level, personal level, interactions with the social environment, and then you have these sort of systemic societal factors that are all interacting, right? And then we can see how these societal factors, social factors are manifesting at the individual level, but we don't often account for all of that heterogeneity because it's difficult to do statistically, right? We can do an interaction, maybe a three-way interaction, but beyond that, it gets very difficult to be able to interpret. But I wanted to try to do that to see, instead of just having these covariates as control variables, or to adjust the models, to actually consider their impact together. So the purpose of the study that I want to share with you today is sort of based on the fact that there are likely particular patterns of relationships between risk and resilience factors that we can actually identify that might help us better understand, one, the equivocal nature of the telomere studies, with some reporting, shorter for one group and longer for another, and then vice versa, to help us really understand the equivocal nature of that literature, and then the conditions under which older adults in particular, and African Americans more specifically, are at an increased risk for telomere shortening, right? And so the goal of this study was to sort of shift the current approach, or conventional approach, to studies of biological aging and race toward sort of a more dynamic and sophisticated representation of individual differences or population heterogeneity, right? And so one of the things I wanted to do was to ask two questions. The first question was, can a biopsychosocial risk typology be identified that explains accelerated aging in African Americans? And that's really looking at biological factors, looking at psychosocial factors, and seeing if, instead of just adjusting for them in models, can we actually sort of look at the heterogeneity and how many of these contextual factors are working together to help us understand, is there a sort of typology, right? So that was one question. The second question, are there modifiable risk factors, right? Is there anything that we can do? Are there any malleable factors that we can intervene on to help to disrupt this process? And if there are they different by race? So very quickly, some methods. I use data from the Health and Retirement Study. I'm sure many of you are familiar with HRS. So I used their first wave of telomere data. There's a second wave now that are available that I'm hoping to get my hands on soon. But for here, these were the data that were available. They were collected in 2008, and it's a salivary telomere length. They're collected from saliva. So the respondents were randomly selected from the larger group of HRS participants, and that N is 5,808. So for this study, we have an N of 5,068 respondents, because we also wanted to include the psychosocial variables, and that was a leave behind survey. And so when you combine these different data sets together, we ended up with 5,068 African American and white respondents. And so the biopsychosocial risk typology, has 6 indicators. Right? So the first is salivary telomere length, chronic health problems, and that's a count, sort of a common count of chronic health problems like blood pressure, diabetes, cancer, etc. Early life adversities, and this is also a count. So people were asked about events that happened to them before they were 18. Everyday discrimination, which is a common measure to assess, different types, 6 different types of discrimination that people are experiencing. This is not based on race. This is just every day. Emotional support. This is akin to social support, right? So we're asking people about how much they feel loved and cared for, and how much they open up about their worries and concerns. And then negative interaction. We're asking about conflict that people have in their network. So these are the 6 measures that were used to empirically define this psychosocial biopsychosocial risk typology. So I use latent class analysis, or LCA, which is just a way of taking into account all of the different interactions that happen between these variables. And what's key about LCA is that it's an individual-based analysis, so it's not a variable-based analysis. It's looking at each individual and their individual response across all of these items, and then creating these profiles as a result of that. And so each profile it's mutually exclusive. Like one person can't be in more than one profile. They're only in one profile or class at a time. I also used latent class multinomial logistic regression. That allowed me to determine, so of these demographic factors and of these psychosocial factors, can we understand who's more likely to be in which class? It's very similar to multinomial logistic regression with just a slight difference, right? So instead of looking at men versus women with, say, men as a referent group, the class is a referent group, okay? So a few results. These are the demographic characteristics of the sample. So of course there's race, there's age, gender, SES, and marital status. And this gives you sort of a general summary of what those demographic characteristics are. There are more whites in the HRS. Average age is 70, slightly more women. And this is the SES, education and income for the sample. And the percentage of respondents who were married, 65.2, versus those who were separated, divorced, widowed, or never married. So this is the bivariate. This is important just so that you can get a sense of who's in which class, right? So when it was all said and done, there were 3 types, right? 3 classes after the LCA was completed. So I labeled them the health risk, the biosocial risk, or the psychosocial risk, and the high risk. And I'll talk a little bit about why they're labeled that way and what they actually look like. But this gives you a sense of how people are distributed in those classes. So what you see here is that African Americans were more likely to be in the high risk class compared to the other classes. In terms of age, the people who were older, on average, were more likely to be in the health risk class. Those who were younger were in the psychosocial risk class. Here are the findings by gender. You see that women were more likely to be in the psychosocial. There were just more women in the psychosocial risk class than there were in the other classes. And men, there were more men in the high risk. Again, key is that men and women are in both, or both men and women are in one class, right? Both African Americans and whites are in the same class. So we're not comparing groups, we're looking at the heterogeneity within the profile. Here are findings for SES. You'll see that the higher income level is in the health risk. I'm sorry, the highest income level is in the psychosocial risk, as well as the highest education. Lowest income is in the high risk. And there's a little bit, and these are statistically different, significantly different. So even though education in the high risk is close to education in the health risk, they're statistically significantly different. And then here are our marital status categories. With people who are married, they're more likely to be, or more represented in the psychosocial risk class, less in the health. And for those who have a different status than married, most of those are in the health risk, with fewer people in the psychosocial. This is just to give you a sense of sort of demographically, how are people distributed across these classes, and what is the proportion of their distribution across these 3 classes. So now this is what the classes look like, right? And so let's see if I can find a pointer here. Let's see, I'm going to use a pointer, that might help. Okay, so for the health risk, that's the blue line. This is on average for this profile. It's 38%, or almost 39% of the sample are in this, what I call health risk. It's really sort of a biological health, where they're high, generally high on chronic health problems. This is their telomere length. It's shorter. It's the shortest of the 3 groups. Here, they're generally higher on emotional support from family, which is great. They're the lowest on negative interaction, which is great because negative interaction is bad. And in terms of reporting discrimination and early life adversities, they're fairly low, right? So generally their risk profile is really determined by their level of chronic health problems and their telomere length. Everything else looks pretty good, right? And then you have this high risk group. That's the red line. They are highest on chronic health problems. Their telomere length is a little bit longer than the health risk, but still it's fairly shorter. And then they're higher on family support, which is really good. Lower, high risk group. They're shorter on telomere length, and then they're lower on emotional family support, which is not good, right? They're also really high on negative interaction. They're also high on discrimination and high on life adversity. So I called them the high risk group. They are really a true biopsychosocial group, right? Where they're high on all the bad things and low on all the good things, right? And so this is the high risk group. And then we have this psychosocial risk group. And the reason I call them psychosocial is you'll see that they're pretty low on their chronic health problems. So they tend to be a healthier group. They have longer telomeres, so that's a good thing, right? In terms of their emotional support, it's sort of in the middle, right? In terms of negative interaction, it's pretty high, right? And then you have discrimination here and early life adversities here. So because of the high levels of discrimination that were reported, the high levels of negative interaction that were reported, and the fairly low levels of emotional support, I call them the psychosocial risk group because their risk seems to be primarily determined by these psychosocial factors, right? Because in terms of their health and telomere length, they tend to fare better than the other groups, okay? All right, so I'm going to walk you through the multiple logistic regression. It is no less awkward in latent class analysis than it is in regression, right? So we'll start with this health risk versus high risk. And so very similar to any multinomial logistic regression, you have a reference group. So for this study, I use the high risk group as the reference category. And so I'm comparing all these factors to the high risk group. So high risk is sort of the high biopsychosocial. Health risk is really just health, chronic health and telomere length. They're pretty much fine on everything else. So comparing these two groups, you'll see that whites were more likely to be in the health risk group versus the high risk group, okay? Interestingly enough, we tend to see findings in telomere studies that really do focus on sort of high, shorter telomeres and health problems, right? And that's where most of our white participants were assigned to, right? This sort of health risk group, not the high risk group. In terms of age, if you're older, you're more likely to be in the health risk group than the high risk group. If you're a woman, you're more likely to be in the health risk group compared to the high. If you have higher levels of education and higher levels of income, you're more likely to be in the health risk group compared to the high risk group. Okay? And there's no effect, well, actually in married and cohabiting, if you're married or cohabiting, you're less likely to be in the health risk group compared to the high risk group, okay? I'll summarize this so that in English, so that it makes more sense. So now we're going to look at the psychosocial risk group. So just as a reminder, these are, this is the profile where most of the risk are psychosocial factors: racial discrimination, everyday discrimination, early life adversities, negative interaction with family, and low emotional support with family. Okay, so for this group, if you compare them to the high risk group, you'll see that people who are older are less likely to be assigned to the psychosocial risk group. You'll see that women are more likely to be assigned to the psychosocial risk group compared to the high risk group. And people who have higher levels of education and income are also more likely to be assigned to the psychosocial risk group compared to the high risk group, right? The group that's high on all the bad things and low on all the good things, right? And so finally, I wanted to compare the psychosocial risk group to the health risk group, right? So the health risk group, again, it's just really chronic health conditions and shorter telomere lengths, and everything else is pretty much okay, right? So comparing them to the psychosocial risk group, we see that people who are older are less likely to be in the psychosocial compared to the health risk. That makes sense because we've already talked about the health risk group being primarily comprised of older people. And also women, they're more likely to be in the psychosocial risk group versus the health risk, right? Just health versus these sort of psychosocial kinds of resources. And the same with SES. So people who have high SES, they're more likely to be in the psychosocial versus the health, okay? And I'm gonna summarize all this because this all, once I started looking at these data, it tended to make sense to me, right? So summary and implication. So I just wanted to summarize that one of the main objectives that I wanted to accomplish with this study was to define these risk patterns, right? It had not been done with telomeres before. It's just, you know, again, we're looking at sort of individual variables and how they might be related to telomere length, but not really looking at patterns of risk. And for me, looking at patterns of risk is really important, as I said, because that's where you can start to find factors on which you might be able to intervene, right? What are those modifiable factors? So we can't change race, we can't change age. You know, it's very hard to change education, particularly when we're talking about older adults, right? So we have to start looking at these other contextual factors that are more malleable, right? So that's one of the reasons that I wanted to do this study. So going back to the first question I asked, can a biopsychosocial risk typology be identified that explains accelerated aging in African Americans, right? So the short answer is yes, right? So again, if there was no typology or different subpopulations in these data, then it would just look like regression. There'd be one class and nothing would be interacting. But we found 3 different subpopulations, meaning these are 3 different groups of people in the same data. And one of the things that we did find was that African Americans were more likely than whites to be assigned to that high risk type, right? So high chronic health conditions, shorter telomeres, low emotional support, high negative interaction, high discrimination, and high early life adversities, right? So African Americans are more likely to be in that group than the other groups, although they're represented in those other groups, right? We also see in terms of demographics that they tended to be younger, married male, and have low SES. This finding is important for me, and I hope important just in general, because it does reveal that the contribution of psychosocial stress in accelerated aging, particularly among African Americans, and it also identifies some modifiable risk factors that we might be able to intervene on to promote telomere maintenance, right? Just to keep them from shortening, and even to promote telomere lengthening. There have been studies that show that telomeres can actually get longer, right? Depending on the types of interventions. So then there's the second question. Are there modifiable risk factors? We saw that. And resilience factors associated with telomere, that's a yes. And if so, are they different for Blacks and whites? That was the second question. So yes, we did find some modifiable psychosocial risk factors, but we also found that for that health risk type, where white participants were more likely to be assigned to than whites, it was primarily characterized by shorter telomere length, multiple chronic conditions, and then a lot of demographic factors, right? So in terms of the psychosocial factors, they were pretty positive, low on negative interaction, high on emotional support, limited exposure to psychosocial stress, both during childhood and during adulthood. It seems to be the health, right, and the demographic factors that were the primary drivers for this particular profile, right? And so since the primary drivers are demographic factors, there are a few really entree points for intervention for this particular group, at least with the variables that were included in this model. And I think it does suggest that we need to look at other factors that might disrupt the relationship between race for whites and telomere length, because these psychosocial factors don't tend to be very disruptive if you're assuming that positive or high levels of emotional support is good and low levels of negative interaction is good. And that's not always the case, right? Because there's a lot of literature, including my own, that shows even optimal levels of support, where you have high levels of positive and low levels of negative, can have detrimental impacts depending on the outcome. And it has to do with sometimes that support is mismatched. It's not the support that people need. It's not the support that people want, right? So it does tend to result in more negative outcomes, right? So we sort of assuming though that negative is bad and positive is good, we might want to look at some other psychosocial variables to better understand what might disrupt this relationship for whites. I want to bring you back to this piece too, because both African Americans were assigned to risk types that were characterized by shorter telomere length. That's important, right? So overall, the literature supports the shorter telomere lengths among African Americans. But what we see here is that's not necessarily the case, right? Both whites and Blacks were assigned to groups that had shorter telomeres. The profiles were different, right? Telomeres, similar levels, but the overall profile was very different. And so this, I think, helps us understand maybe why some of the findings might be disparate, keeping in mind that we're talking about different samples, different measures, right? So obviously we're going to find some differences because of that, because of different methods. But it does really highlight the importance of looking at heterogeneity, because with two groups with shorter telomere lengths that look very different, right? Other different factors to consider is definitely a contribution, and it sheds light on how telomere length can reflect genetic, environmental, and psychosocial factors that can vary substantially across groups. The psychosocial.
I hate to interrupt, but we'd like to leave a little time for Q&A.
Yes, very good. So I just want to add here that the psychosocial risk type was the only profile that was characterized by longer telomeres, which is good, right? So it does, African Americans were slightly overrepresented, but I do want to highlight the fact that race did not determine which group would be assigned to this profile, right? So that's important to consider as well, that longer telomeres generally are reported by race. There were no racial effects, but there were other factors to consider to help us understand why there might be longer telomere lengths. In general, this was a healthier population. And then there's also, one really quick thing I wanted to say is that there is this paradox that is the other reason that I wanted to do this study. And that is because there are studies that show that for people who experience extreme stress or acute stress, their telomeres lengthen, they're longer, right? And I thought, well, if people are getting surveyed at that point in time in their life, then they're going to have longer telomere lengths, which typically is associated with better health. The reality is, in this case, it might be a result of oxidative stress, right? So I think we really need to start poking around a little bit more, and longitudinal data will certainly help with that. But again, longer telomere lengths don't necessarily reflect a healthier population. It might be something else that's going on that we haven't discovered yet. So here are my take-home points. I'm going to leave them at the end. And then this study is published already in the Journals of Gerontology, Medical Sciences, and it was just published this year. And then future research, lots of future research suggestions for telomere length. I'm going to move on to really quickly talk about this study that I just got funded. I'm going to be working with Jude Carroll and Steve Cole, who is the pioneer of social genomics, to look at racial discrimination and gene expression. So we're not going to be looking at telomeres. We're going to be looking at all different types of neuroendocrine and sort of cellular replication, transcription, genetic factors to understand how racial discrimination gets under the skin. All right, very good. That's all I got. Thank you.
Thank you so much, Karen. That was fantastic. We're going to now open it up for questions. And of course, the first two go to graduate students. And so you can either put it in the chat or just jump right on in.
Please don't put them in the chat.
Okay.
I'm horrible. If they do, you can read them to me, Bridget.
Of course.
Thank you.
I got you. Okay. Graduate students, where are you? Well, while they're formulating their questions, I am curious. So thank you for doing this. This is interesting around accelerated aging. Part of me wonders though, because we think a lot about psychological outcomes as being independent of the physical health, when they are pretty intertwined. And I wonder how you think about the possible links between the psychosocial and why you might see. Just talk a little bit more about why the psychological might be showing up in terms of, you know, affect and mental health outcomes, but not the physical aspects of manifestation. Might you see something different if you were to look at different biological markers as opposed to telomeres? Like, I'm just curious about how you're thinking about that.
Well, you know, I can only speculate. That's a good question, Bridget. So one of the things that I have been looking at is just how we respond to stress, right? So, you know, you can take James Jackson's affordances framework, particularly with African Americans who might respond to stress in ways that might be more harmful for our physical health, whether it's, you know, what we eat or, you know, what we consume or not do in terms of physical activity. We might feel better psychically or psychologically, you know, and that might reduce our risk for mental health disorders, but it might increase our risk for hypertension. And we have evidence to show that. What I'm hoping to do with this new study to look at the genetic level, because we don't know. There just aren't enough studies that really help us understand how African Americans, and this is a study of African Americans, by the way, we're going to collect blood and sort of look at gene expression to see how people are responding to that. And that's of interest to the UCLA Cousins people. I injected, when I proposed the study, to look at cultural mistrust, which is a psychological variable, which might help us understand why we might see genes expressed in a certain way based on racial discrimination in some people and not others. And it's just my first attempt of looking at the psychological variable to see if we can sort of find this intervening factor, and hopefully we'll see that we can actually disrupt that process, right? People who are experiencing racial discrimination, there's actually something that we can do in the middle there so that it doesn't result in these poorer health outcomes.
Yes, we should talk more about this because this is fabulous. I'm excited that you're doing this. So
I'll send you my proposal after we finish.
Right,
You're welcome.
Mark has a question.
Well, I have too many questions actually. Hi, Karen.
Hi.
Talk. I was thinking about when you talk about the length of telomeres with respect to stress and how acute stress may affect the longer in terms of telomere and stimulate growth. So it actually made me, that one comment made me think about the nonlinearities and associations between telomere length and various kinds of indicators that you're using. So, you're really assuming a lot about linearity in terms of these relationships. You made me think about non-linearities where as you move through the distribution of some variable like stress, but then you get to really acute stress, then you begin to learn something about telomere length. So it's just a suggestion about how to probe the non-linearities of the telomere issue.
Mm-hmm.
The question I have though is, it looks like, I'm just going to ask, are you using the epigenetic clocks? I'm thinking what Eileen's done with Morgan Levine and in terms of that work, and using the epigenetic clocks to kind of compare and contrast with what you've done with telomere length. So there's all that work on biological aging from that perspective, and I was wondering if you were to replicate your analysis, or if you were to use a different measure, what would you expect to get the same results? Because it's really different ways of marking biological aging. So
right. And it's a good question, Mark, and thank you for that. And I imagine yes, right? Because I think, looking at mitochondria and the epigenetic clock, and I'm super excited to work with Steve because that's what they do over there. And that's some of the things that we'll be doing, is sort of looking at the genetic level and all these different markers to see which ones sort of rise to the top. Right, and I think in the combination of them, and so we're sort of documenting what it might look like at the genetic level, and just to get a basic understanding of what different stories that they might be telling, depending on the type of stress that people might be experiencing.
You might think about also looking at, Eileen and Morgan have published some work on cognitive age, like brain age, and do some comparisons with brain age versus biological aging, because they're not the same, and to get a handle on some of these race issues with respect to well-being, dementia compared to other kinds of disease experiences in older age. Just a thought, awesome. Because you've got some good material, and Steve's lab's great at UCLA. So.
Well, thank you. Well, real quick, Mark, thank you for that. Thank you for that too. And also real quick, I have a study under review at NIH looking at perivascular space and cognitive impairment with African Americans. Yeah. We're going to be looking at that.
Great. Good. Congratulations.
Thank you.
You're ahead of the curve. That's good.
No, not yet. But thank you for that.
So Rebecca Wong has a question in the chat. Rebecca, did you want me to just read it out, or? So, go ahead.
Yes, Bridget, go ahead. Thank you.
Sure. So, so she had two questions. The first was for the future wave of HRS TL. Will you be able to conduct this in Hispanic groups? And then her second question was, the mean sample is around 70 plus years old, which seems old. And this was because you got saliva, is the question. And can you comment on the differential survival of African Americans versus Hispanics in terms of the age, and how this could impact your findings?
So I don't conduct a lot of research on Latino populations, and I certainly can't speak to HRS. I think one of the reasons why I wanted to just focus on African Americans and whites is because of I wanted to speak directly to some of the findings that have been reported. One, two, the sample of Latinos and HRS is not great. And so it's, and I'm not sure what it's going to look like for this second wave of telomeres. I don't imagine it's going to be any better in terms of the numbers in order for us to be able to examine ethnicity and as well as race. So unfortunately, I can't speak to the HRS. In terms of, what was the second question?
About
the age and the
age and the type of collection to get the telomeres.
So in terms of, they're collecting telomeres differently, the second wave. I'm not sure how, they're not using saliva for the second wave. So I'm not even sure if they're going to be able to legitimately compare saliva to blood, and they're using a different lab. And so that's a whole nother situation. And in terms of the age, you know, there's lots of discussion about like who's left. Like if African Americans are dying earlier, then who's left? Are they healthier? Are they more resilient? You know, is there something else that's actually happening? Is there selection bias? Right. And so yeah, there's always going to be that concern. We can't control for everything. However, someone who works with African Americans every day, older African Americans, there's still a lot of people with a lot of variation in terms of really healthy and not so healthy who live to be, you know, at older ages. And so I do think that there's still enough variation for us to make some definitive conclusions with respect to age.
And so I think that we're going to need to wrap up if there are no additional questions. But again, I want to just reiterate how much we appreciate you sharing your amazing work with us today for us, for the PRC and the CAPS. Thank you so much, Karen. And we look forward to seeing more of your work. Thank you.
Thank you. Thanks so much for having me. I appreciate it. Thank you. Right. Happy Friday, everyone. Have a wonderful
Happy Friday.
Thank you.